Now we are going to look at how PCR has impacted health in detecting different pathogens and also quantifying how much pathogen is present in a sample. Say someone is infected with a virus. Initially, detecting a virus was a big challenge because the initial technique, which is called ELISA, was dependent on the amount of protein present in the sample. So very low quantities of virus could not be detected. Because we need enough protein to be able to detect it by ELISA method. The power of this technique, PCR technology, we can amplify one strand of DNA and make billions of copies of that particular strand. And it is easy to detect even very low amounts of pathogen which are present in the sample. Say, for example, a patient has a virus in the blood. We remove the blood from that patient. We isolate the plasma, that is the non-cellular part of the blood, the liquid part of the blood. We take a small portion of that plasma. We extract the DNA out of that sample. If the virus is present, we will have the viral DNA in our sample, extracted sample. We can run a PCR reaction of our sample. We know the special sequences of the virus, so we can make the complementary primers for that particular virus. We run the PCR reaction. Now, if our sample had virus in it, its DNA will be amplified, and we also know what size fragment would be generated by this PCR procedure. When we run the gel, if the band is present, our patient has the virus in its blood. This technique is being used to detect hepatitis C virus, hepatitis B virus, HIV, which is human immunodeficiency virus, adenovirus, influenza virus, cytomegalovirus, and also several other pathogens, bacteria also. Here we have been able to determine whether the virus is present in the blood or not. The answer is either yes or no. Physicians want to know more. For example, a person is suffering from HCV, hepatitis C virus. A physician puts that patient on a drug therapy. Now the physician or the doctor wants to know if this particular drug he is giving the patient, whether it is working or not. Meaning whether the amount of virus in the blood is decreasing or not. How do we do that? That is quantitative PCR. It is also PCR. It's the same PCR we have just seen. However, it has a special molecule in it. It has a little primer. I will highlight this primer. Here is the primer. This primer is called a probe because it is not going to be extended like the other PCR primers. This primer, now I'm going to start calling it probe. This probe will bind DNA, the target DNA, or the target DNA being our viral DNA, at a specific location. When the PCR reaction is run, this probe will be degraded. This probe has two important molecules attached to it, R and Q. R is the reporter molecule which glows under certain light conditions. Q is the quencher molecule which will absorb all the light being produced by the R molecule. These molecules will, if they are adjacent to each other, they will not produce any light. But if Q and R are separated, the light will be produced because quencher is no longer in the vicinity of the reporter. It will not be able to absorb the light of the reporter and light will be produced. So here's our PCR reaction. We have the primer 1 and we have the primer 2, and we run our PCR reaction. If the virus is present in the sample, the DNA polymerase will extend. When it extends this primer, it will degrade this probe. When this probe is degraded, the quencher will be separated from the reporter. They will go their separate ways, and reporter will emit light. The amount of light is directly proportional to the amount of sample or the viral DNA that was in the original sample. So if our sample, say for example, had only one viral DNA, only one photon of light, which is a unit of light, one particle of light will be emitted and it will be detected. Let me show you in a different way. Here is our quantitative PCR. Here's the target DNA. Here is the primer. Here's the DNA polymerase. Here is our probe with the quencher and the reporter next to each other. This reporter is not glowing because the quencher is absorbing all the light. When this PCR reaction is run, the DNA polymerase in this case will degrade the probe and separate the two molecules, the reporter and the quencher. When the reporter has been released from the probe, it will emit light. This PCR machine, which is used for quantitative PCR, has a very sensitive camera in it. It can detect very minute amount of light. So now if we originally had one viral DNA in our sample, one light particle or one photon, let's say for the sake of argument, will be emitted. And not only this camera can detect it, it can also quantify it. It will know how many photons were produced. If there were 10 viral samples and 10 viral DNA fragments present in our sample, 10 photons will be produced. 100 photons will be produced. So this technique not only allows us to detect the virus, it also allows us to determine how many molecules of pathogen were present in the original sample. This allows physicians to determine whether a particular therapy against a virus or a pathogen is effective or not.